Suppose there is a cell. In this cell there is a nucleus. In nucleus there are so many genes which are kept in inhibited state. Adverse condition came, toxic stimulus came. We know that this cell can destroy this toxic stimulus. But this gene is present in the form of heterochromatin. So this heterochromatin will be converted to euchromatin, will synthesize messenger RNA. Messenger RNA will go to cytosol. This messenger RNA will make a protein. Protein will go and will destroy toxic stimulus. Nothing important. I told you the same thing as previous. How this is done? Whenever toxic stimulus come, DNA is activated, means euchromatin is formed, synthesize messenger RNA. Messenger RNA go to cytosol, protein destroy the toxic stimulus. Function is done by protein. I have already told you multiple times, RNA can't do function, DNA can't do function. If you want to do function, you must synthesize protein. So protein destroy this toxic stimulus. Now what happened? This toxic stimulus came. Listen carefully. This toxic stimulus came. This gene is activated. Synthesize heteronuclear RNA or messenger RNA, go to cytosol. It was about to synthesize protein. Protein is not synthesized till now. This messenger RNA was about to synthesize protein. This toxic stimulus is gone. ट्रांजिएंट था, इट वाज ट्रांजिएंट. स्टिमुलेट किया, भाग गया. कहते हैं ना छेड़ के भाग गया, तो छेड़ के भाग गया. यस, स्टिमुलेट किया, इट स्टिमुलेटेड एंड रैन अवे. ओके, रैन अवे. नाउ व्हाट शुड दिस मैसेंजर आरएनए डू? What should messenger RNA do? Messenger RNA should synthesize protein. Yes or no? Please do reply. What should messenger RNA do? When this toxic stimulus is gone, this messenger RNA should not synthesize protein, because if messenger RNA is lying in the cytosol, if messenger RNA is present in cytosol, it will not affect. If messenger RNA is lying in the cytosol, it will not affect the cellular function. But this messenger RNA, if synthesize protein, this protein has function. I told you that function is done by protein. If this messenger RNA make protein, then this protein will search where is the toxic stimulus. Just like Mukembo, press the button of a missile, this missile will go and will search where is the target. When this protein will not get the target, it will affect the cellular function. So we want that when this toxic stimulus is gone, we don't want protein synthesis. So how you will inhibit protein synthesis? How this messenger RNA will come to know? Again I repeat, if this messenger RNA remain in cytosol, we are not concerned. But if this protein will formed, this protein will disturb cellular function. Yes, every protein has its own half-life. It will disturb the cellular function during this whole half-life. So what will happen? If this toxic stimulus is gone, then microRNA will come. What I said, microRNA will come. This is microRNA. MicroRNA will bind to messenger RNA at which end? 3 end. MicroRNA bind to messenger RNA at 3 end. Yes, question was asked, 3 untranslated region. Which end? 3 untranslated region. Yes, microRNA bind to messenger RNA at 3 untranslated region and inhibit protein synthesis. How it will inhibit protein synthesis? First, either degrade messenger RNA or remain attached to messenger RNA and not allow protein synthesis. Okay, so microRNA bind to messenger and it bind to 3 untranslated region, inhibit protein synthesis. First, either degrade messenger RNA or remain attached to messenger RNA, not allow to protein synthesis. In any case, this microRNA will not allow this protein synthesis. This is the reason that it is interfering the messenger RNA. That's why this is called RNA interference. Yes, mechanism involved in RNA interference is gene knockdown. This phenomena is called gene knockdown. What is gene knockdown? Gene knockdown means down regulation of gene. Down regulation of gene means this gene will not be expressed. Down regulation of gene knockdown. Okay, clear? So RNA interference or yes, microRNA is same. RNA interference, microRNA, mature functional microRNA, RNA induced silencing complex, small interfering RNA silencing RNA. All are same. Again I repeat, RNA interference, microRNA, mature functional microRNA, RNA induced silencing complex, small interfering RNA, silencing RNA, function is same, that is gene knockdown. Remember this point. What is the difference? Small difference is there. Yes, in the structure, how they are produced. Yes, how they are produced, but that is different thing. For the time being, in any question, if you find silencing RNA, small interfering RNA, RNA induced silencing complex, yes, you can easily understand. This is small interfering RNA. This small RNA, very small RNA, interfere with protein synthesis. Silencing RNA, it is silencing the messenger RNA, means it's not allowing to protein synthesis. Small interfering RNA, because it is interfering with the protein synthesis. RNA induced silencing complex or mature functional microRNA, all are same. They inhibit protein synthesis from messenger RNA when not required. Gene knockdown, down regulation of gene, both are question. Now, why we are so much concerned about microRNA? Every time they are asking question, what is the reason that they are asking question again and again? The reason is that proto-oncogenes is converted to oncogenes. Yes, oncogenes means this will give rise to this will give rise to cancer. Suppose I came to know there is a person having cancer, and due to this over expression Means these oncogenes are synthesizing more and more RNA and more protein. Now I thought that I can treat this patient. I know that this patient is having this. I know this patient is having cancer, and this cancer is due to overexpression of this proto-oncogene. I made microRNA for the RNA of that proto-oncogene, and that proto-oncogene will be inhibited, protein will not be produced, cancer would be treated. Got the point? So this microRNA is topic of research. What is untranslated region? I have not pointed out. Otherwise, whenever you will see the messenger RNA, you will find a small portion of RNA which don't take part in protein synthesis. Those are called untranslated region. Untranslated region means the portion of RNA which don't take part in protein synthesis. Untranslated region. At that place, it bind. So this is microRNA, RNA interference, how they are produced I will discuss, don't worry. For the time being, make a mnemonic. If microRNA questions are asked, and they usually confuse in this, gene knock in, gene knock out, gene knock down. So simply you can easily remember this is 3 untranslated region. Okay, gene knock down, gene knock in, gene knock out. Knock in means you will add gene in the DNA, gene knock in. Gene knock out, you will remove gene from the DNA, gene knock out. Means you are working at DNA, knock in, knock out. But if you are taking care of RNA, gene knock down. If you are inhibiting RNA, gene knock down. So mnemonic is microRNA M. Rotate once, 3 untranslated region. Rotate twice, W. So in, out. Only down is having W, looking like M, opposite of M. So you will remember at least in exam, rotate once 3 and rotate twice W. Okay, understood? So this is RNA induced silencing complex. So again I repeat, there was a primary microRNA gene, RNA polymerase II synthesized primary microRNA. This primary microRNA is acted upon by DROSA nuclease. DROSA nuclease cut cap, poly A tail, DROSA, Pasha, Jadugar. Huh? You will remember. Now, yes, primary microRNA is going to pre-microRNA. Pre-microRNA come out of the nucleus. Pre-microRNA form microRNA duplex with the help of dicer nuclease. Dicer cut from the end. Dicer cut from the end. Cut from the end and form microRNA duplex. This microRNA duplex, look at me, this is microRNA duplex. Yes, now I told you how this microRNA is produced. Yes, we found that in our nucleus primary microRNA gene is there. So I remove everything just to write again, okay, so that you can understand. In gene, in that, yes, in nucleus there is primary microRNA gene. With the help of RNA polymerase II, this primary microRNA is produced having cap, poly A tail. Then what happens? Then DROSA nuclease comes and it cut this end, cap and tail, and primary microRNA is converted to pre-microRNA. Pre-microRNA comes out of the nucleus. Pre-microRNA comes out of the nucleus. Pre-microRNA is acted upon by dicer nuclease. Acted upon by dicer nuclease, cut this to form microRNA duplex. microRNA duplex. This microRNA is disulnuclease cut this end and this form microRNA duplex. Now microRNA duplex, yes, these two strands separate. That is called unwinding. Strand selection. This is unwinding. Strand selection. Out of these two strands are separated. One strand is discarded. Another strand is attached with protein. And when now this is called RISC, RNA induced silencing complex or mature functional microRNA. So this is the way how microRNA is produced by microRNA, primary microRNA gene with the help of RNA polymerase II. This having cap, poly, tail, drosonuclease cut cap and tail. Primary is converted to pre come out, pre microRNA is converted to microRNA duplex by enzyme disulnuclease. Now this microRNA duplex, there is unwinding, strand selection, one strand is discarded, another functional, it bind with protein to form mature functional microRNA, or another name is RNA induced silencing complex. So this is microRNA and the function is to gene knockdown. And now we are, yes, we are more and more questions are being asked. The reason is that if we want to suppress any gene, we know that many genes are overactive in many cancer, so we can treat those cancer by inhibiting those genes, not at the gene level but at the RNA level. We can inhibit the protein synthesis, oncogenes protein, by inhibiting the messenger RNA. Understood? So now this is clear to you. Now we are left with protein synthesis, genetic techniques,